Hi guys, so today we're doing the most high yield topic in biology for HSC, uni, and medicine. That's mitosis and meiosis. And I wanted to explain the most important things you need to know to answer any question and break down the processes very simply. So I'm going to tell you everything that just matters in mitosis and meiosis. So we're going to start with the big picture, and that's just the differences. So with mitosis, I just like to remember MIT for mitosis. It's occurring in all body cells or somatic cells. It's important for growth and repair. And a sentence that I just like to lock in into my memory is that mitosis outcomes or the products is two genetically identical diploid daughter cells. That's what we're yielding. Two genetically identical diploid daughter cells. When we look at meiosis, that's like meiosis. It occurs only in the sex cells or the gametes. It's important for the continuity of life, sexual reproduction, and this one we're going to yield four genetically unique haploid daughter cells. Four genetically unique haploid daughter cells. I just like to remember these because you can answer any comparison question and just secure any marks that you can by putting all that information into one dot point. Okay, so what does haploid and diploid even mean? Okay, so when you usually see N will stand for the number of chromosomes set. An image here that I've drawn is we can see a chromosome and we can see one chromatid and another sister chromatid. They're joined at a centromere, and two of these chromosomes, we've got one chromosome from mum, one chromosome from dad. Both of these make up a chromosome set. A woman, for example, will have a chromosome from the mum, from the dad, and then that will combine with another chromosome set from the male's sperm, who has from his mum and his dad, and then together those will create a diploid cell, or the zygote. All right, so now let's just quickly go into the processes. So I like to remember the cell division through just saying PMAT. So P, prophase, M, metaphase, A, anaphase, T, telophase. Before mitosis or meiosis begin, we start with our interphase. That's where the cell's at rest. There's DNA replication happening. So we're just we're just duplicating the amount of DNA we have. Okay, so let's start with mitosis. That begins with prophase. In prophase, we have 2N. We're diploid. We've got two chromosome sets. And in prophase, our chromatin condenses and our centromeres form and start to move to the poles. Then we've got metaphase, and the most high yield thing you need to know about metaphase is M for middle. So we're lining our chromosomes at the middle plane. So at that metaphase plane or the middle plane, we're lining up here, and we're lining up in like a single file. That's how most pictures will appear. And we've got some of these spindle fibers coming out of the centrosomes, joining at the centromeres. Okay. Then in anaphase, remember A for apart. This is where the spindle fibers will contract and they will pull the chromatids apart. So A for apart, anaphase, we're pulling it apart. And here you can see that we still have the same amount of chromosome sets, but we're just moving the chromatids around. That's why in cytokinesis and telophase, we result in diploid. Okay, so two chromosome sets remain. In telophase, our nuclear envelope reforms again. And in cytokinesis, cyto for cytoplasm, we're splitting the cytoplasm and we're resulting in our two genetically identical diploid daughter cells. Okay, now let's move on to meiosis. And this is going to be really simple. The only thing that's different here is that PMAT happens twice, and the first time it happens, we've got genetic variation processes happening. And also in meiosis 1, you've got reduction division happening, and we'll go through that. Okay, so prophase 1, again, we start off with our diploid cell. Our chromatin is condensing after we've just done interphase, and something that important happens here is crossing over. You just need to remember this all the time for all your exams. Crossing over will occur between homologous chromosomes. Homologous chromosomes basically mean you have the same size, shape, and genes in those chromosomes, and they will cross over and exchange genetic material here. That only occurs in prophase 1. It doesn't occur in prophase 2. Okay, metaphase. Again, M for middle. We're lining at the middle plane, but here we've got tetrads lining. So tetrads meaning like 4, and we've got 4 chromatids and 2 chromosomes that are homologous lining up together. And we've got our spindle fibers forming, our nuclear membrane degrading. That's all in metaphase. Anaphase, we're pulling the chromosomes apart. We're not separating the chromatids, we're actually pulling chromosome sets apart. That's why we're reducing how many chromosomes we've got. So we're ending up with a haploid cell because we're separating those chromosome sets. And here, this can happen randomly, okay? So this can segregate here, that can segregate there. This is where random segregation occurs. That's another source of genetic variation. So then we've got telophase, our nuclear envelope is reforming, and cytokinesis, we're splitting. So here we've ended up with haploid cells because we've split apart those chromosome sets. Remember, haploid and diploid only refer to sets of chromosomes, not chromosome number, not chromatids, only chromosome sets. Okay, and because we've lined up at tetrads, we've pulled the chromosome sets apart. Okay, then our meiosis 2 is basically like mitosis. We're doing everything very similarly, and we've got chromatin condensing. So now this is happening in 2 cells. Chromatin is condensing, centrosomes form at the poles. Metaphase, we're aligning. Now we're in single file at the middle plane for metaphase. Now we're pulling apart in anaphase, A for apart, and telophase and cytokinesis 2 happen. But now we've ended up with 4 genetically unique haploid cells to finalize. So we can see that we've got those crossed over chromosomes here. And yeah, so that finishes our mitosis first meiosis, just everything you need to know. Thank you.